Welcome to Mr. Chalk's Revision Tips. In this video, we will look at
the neuromuscular junction, muscle contraction and depolarization. So, according to the sliding filament theory, the actin, which is the thin filaments of muscle fibers, slide past the myosin, which is the thick filaments, during muscle contractions. So the contraction is triggered by a neural impulse which reaches the muscle at a neuromuscular junction. So an action potential moves down the axon of a motor neuron towards the neuromuscular junction. When the action potential reaches the neuromuscular junction, it stimulates calcium ion channels to open. Calcium ions diffuse into the synaptic knob, where it stimulates synaptic vesicle to move to the cell surface of the neuron. The neurotransmitter acetylcholine is secreted from the neurotransmitter vesicles into the neuromuscular junction by exocytosis. The acetylcholine binds to specific receptors on the postsynaptic membrane, which is called the sarcolemma, causing sodium ion channels to open. Sodium ions diffuse into the sarcolemma, causing depolarization. The bound acetylcholine is broken down by acetylcholine esterase into acetate and choline. This is then reabsorbed by the motor neuron, where it can be recombined back into acetylcholine. The depolarization of the sarcolemma travels into the muscle fibers, spreading through T tubules. So these are in contact with the sarcoplasmic reticulum. When the action potential reaches the sarcoplasmic reticulum, it stimulates calcium ion channels to open. The calcium ions diffuse down their concentration gradient, flooding the sarcoplasm with calcium ions. The calcium ions bind to troponin, causing it to change shape. So, what we now need to move on is to think about the physical sliding of the filaments. So, muscle contractions requires a large quantity of energy. So this is provided by the hydrolysis of ATP into ADP. The energy required for the movement of the myosin heads and to enable the sarcoplasmic reticulum to actively reabsorb calcium ions from the sarcoplasm. So the first thing that happens is that the myosin head pulls away from or pulls on the tropomyosin, moving it away from the actin-myosin binding sites on the actin filament. So now that the binding sites have been exposed, the myosin head binds to the actin filament, forming an actin-myosin cross bridge. An ATP molecule can now bind to the myosin head. This causes the head to detach from the actin filament. So the calcium ions present in the sarcoplasm also activate the ATPase activity of myosin. This hydrolyzes the ATP to ADP and phosphate, releasing energy which the myosin head uses to return back to its original position. So the myosin head can now attach itself to another actin-myosin binding site further along the actin filament, and the cycle is repeated. So the cycle continues as long as the muscle remains stimulated and moves the actin in the opposite direction to the myosin, which in turn causes muscle contraction. So if we look at what this looks like, so according to the sliding filament theory, the myosin, which is the thick filament of the muscle fibers, slide past the actin, which is the thin filament, during muscle contraction. So while these the two groups of filaments remain relatively constant length. And you can see from the diagram below, there are a few key features that if you're drawing this or asked to identify, you need to be able to recognize. So we've got the myosin filaments, we've got the actin filaments, there's a part called the Z line, there's an M line, and there's an I band and an A band. And you can see there that the I band and A band change lengths during contraction.
Thanks for watching.